So I'm going to start, and we're going to switch to me. And every year, I try to provide a 30,000-foot view of the changing topic of immunology from a clinical immunology perspective. And, you know, the message is really the same, but I try to tell a different story each time, and I'm clearly going to change the story today. And for those that struggle, like all of us, to, you know, kind of maintain our 30,000-foot view and understanding of basic immunology, there's a million books. I do like the Boss book. It's now in its 6th edition, and it's very approachable, and it's a keeper for certain things. But in the field of the internet, we always have access, because every day you see something that is unfamiliar with you. All of our financial disclosures are well published in this, but these are my real disclosures. So we're engaged in medical Twitter. We're engaged in rheumatology Twitter. A lot of my faculty here is tweeting this out, even as I see it. I can see Adam Brown over here. He's already probably sent out 4 tweets. But please join us. It's kind of fun. We don't have a hashtag for this. We're just kind of doing this back and forth. But my Twitter feed is all things that are immunology, all things that are COVID, and intermixed with placebo and the science of empathy. Helio is here to cover this. Many of our advisors for Helio are here. It's a great publication, and thank you for joining us. So what am I going to do today? I thought I would do this differently. So I have always kind of gone through this immune response in a canonical way, and now I want to change it, and I really want to talk about some stories. I mean, understanding the field of immunology today is kind of like this. This is, you know, I really feel bad for people that are kind of coming into clinical medicine right now. You know, as I look here and I see Dr. Rigby and Dr. Linney over here, I mean, when you're first back and young, energetic guys and walking into the lab, and there was some kind of project that was going to involve something like a T cell or a B cell, whoever the professor or the mentor was could probably draw the entire schema of immunology on a yellow sheet of paper. Said, yeah, there's a stem cell, and it goes to the T cell, the B cell, and then maybe it makes a few things that mediate this. And there's a thing called complement that's really complicated. We'll save that for later, you know. Today, we live in a world that has accelerated at an unforeseen pace. And to pick up the current literature in immunology is to be confronted with, you know, big data. And big data can look like a million different things. And, you know, not only do we talk about cells, we talk about clouds of cells. And to actually understand this, we use integrative bioinformatics that project this in far beyond 3 dimensions, and we use these dimensional reduction analyses. And if this is what you're trying to understand without understanding the story of immunology, why it's here, why we think it is functioning to protect us and to enrich us, to live lives that are worth living, it's a tough one. So I look at it as this. I use this for a lot of different talks, but you know, I was just watching the thing about the James Webb Telescope and seeing the new galaxies out there that they never thought were there. This is the immune system. And, this is, it now has the vastness of big data. This is looking at, literally trillions of data points. But what your eye catches are these big galaxies. These are kind of concepts. You're not looking at the vastness in the background and the darkness between these. So we have a lot of things in science that are like immunology. If you were talking about basic cardiovascular biology or neurobiology, somebody would be giving this same talk. And, regardless of whether you're trying to make this reductionist or expansive, functional, or, just purely descriptive, the best way to understand what's going on in immunology is to understand it through stories. Because that's how we understand the most complex things. And so I'll tell a few stories. We'll pick a few things out, and we'll see how this goes. So the story, if we go back to Hippocrates, this is, the way medicine was devised. The concept of immunity, the concept of immunology was, certainly immunology was not with us, but even the word immunity, which comes from the Latin immunitas, which meant exception, exemption from serving in the military, exemption from having to be fealty to a church or state. That's what it was. There was no idea of disease. And even up through 1,000 years later, the notion that one infection could protect you from another was just kind of an ephemeral thing. Hippocrates posited that we understood everything with the four humors. This was picked up on the right by Galen, the physician to the gladiators. And this canonical science existed for 40 generations. And nothing changed. I mean, we went through the Dark Ages and even through the Renaissance. And, this is puking and purging and blistering, and this is the way medicine went. It really wasn't until about 500 years ago that the notion that medicine was a science based on anatomy, Vesalius, who threw away the Galenic canon, that there were actually connections between the heart, and that's how blood was moving around. He actually defined this. Harvey, the first element of physiology into medicine was introduced, and he was considered a heretic. And he always traveled with a scabbard because he was being attacked. It's kind of like in the COVID era, Dr. Fauci has to have bodyguards. And on the bottom, perhaps the greatest innovation was Morgani, who actually said that disease is different than life. And he studied, thousands of cadavers and wrote his magnum opus, De Sedibus de Morborum. And so now we had anatomy, physiology, pathology. But where's immunology in this? As I said, there's some just little glances that in certain plagues, which no one really knew what the nosologic entities were, that if somebody had it for one time and they recovered, they're probably going to do okay the next time. And that's what was captured. If we move now just 2 centuries ago, all of this basic science, if you will, had very little to do with the patient. The patient would come in and someone would, grab them and feel a pulse, and they might look at their urine under the light uroscopy and that's how they decided whether anybody was sick or not. It wasn't until the development of, physical diagnosis, in the sense that we could actually look at somebody and examine them and know what's going on inside of them, that brought, excitement. And Leonard, in the earliest 19th century with the stethoscope, and Arnbrugger, who worked out the science of percussion, that he could feel that fluid, and he was son of an innkeeper, and the reason that he figured this all out, because as a boy he was always sent down into the basement to figure out how much beer was left in the kegs, and instead of dipping it in, he figured if he could tap on that keg, he knew that it was half full or half empty, or whatever. It's a great story. But where is immunology in this? Immunology is nowhere in this. Even as I show moving forward, the late 19th century, when all things moved from France to Germany, the laboratory-based developments of looking at chemistries of urine and blood, immunology was still missing in action. So if we think of the beginnings of immunology, and most people would go to Jenner in the 1700s. The notion of vaccination, variolation was alive, even though they knew that exposing a person to a virus that could protect them from a subsequent virus, and he obviously didn't know what it was, a virus, it was just a putative infectious agent, there was no underpinning of a science that could explain this. None. And even if we go then to Pasteur and Koch, who refined the notion of vaccinology, there was no scientific underpinning. There was no physiology. There was no anatomy of the immune response. It was just an observation. Pasteur thought that his vaccine was competing for nutrients with another infection that would come along. I mean, it was not there. It was not till nearly the beginning of the turn of the 20th century that we have all lived in, that a scientific underpinning of immunology was born. And here is Emil von Behring, the winner of the first Nobel Prize in medicine and science, and for his, land-breaking discoveries and work, and the commercialization of serotherapy. And he knew that there was something in that serum that could protect a person from a subsequent infection. So it was all about serotherapy. And to fast forward this, the remarkable and romantic elucidation of this by Ehrlich, and these photographs, or these drawings, that the way that he viewed this, cells and these amboceptors, these antibodies that could bind to a cell surface and modulate them. And, it certainly had flaws, but this was before you could see these cells and molecules. He had a vision that was extraordinary. And without Ehrlich, von Behring never would have been successful, because he actually quantified the science of serotherapy to make it safe and effective. All right, so that's just an introduction to when immunology was born. If I had more time, I would throw in Metchnikoff, because he was the guy that balanced the serotherapist with the cellular therapy. But this is not a talk on that. And in fact, if anybody goes to CCR East this year, I'm giving a talk on the history of immunology, which is the real depth. I'm just giving the 5-minute part. So what is immunology today? Immunology is a complex science. It's a field that we believe that protects us from danger, that allows us to discriminate threats that are pernicious from those that are passive. And it is an expansive field that not only protects us, but is involved in tissue homeostasis. It is constantly involved in repair for us to maintain health. This paper, this narrative review on humoral innate immunity and acute phase proteins, which just came out this month in the New England Journal by Mondovani, I found this a very compelling review, so I recommend it to everyone right now. And it kind of looks at the field of immunology as being much more fundamental in, maintaining our health and protecting us from pathogens, the way that we often reduce this. And down below, we see this classical drawing of the acute phase response, which my good friend, Dr. Irving Kushner, published in the New England Journal decades ago. And that we need this system to function at many levels. So the immune system is all about balance. This is a figure that Xavier Marriott and I published several years ago, talking about immune-related adverse events from checkpoint inhibitor therapy, and how when we give checkpoint inhibitors, we tilt the scales, we reinvigorate people's immune system to kill the cancer. At the same time, we develop these immune-related adverse events. So on one hand, our immune system is, enriched with these effector pathways of cells that can kill, cells that can mediate inflammation, cytokines, integration of both innate and adaptive immunity. And on the other side, there are these tolerogenic and regulatory signals that are both soluble and cellular that maintain this homeostasis. So the whole job of the immune system is to protect the host from a danger, whether that be an infection or a dysmetabolic state, and at the same time, not to harm us. And I'm going to use COVID-19 as a kind of a model of how this actually works moving forward. So this is COVID. I thought this is the way that we should think about the immune system. It's very funny. A few years ago, if you told somebody at a cocktail party you were an immunologist, like, who could care less? And what's that? Now everyone's an immunologist, and you're now being quizzed, and so it's kind of interesting. So this is the model that Cassie and I have used for a starting point to kind of understand this, and we'll be talking more about it tomorrow morning. And here you have the natural history of COVID. So, you're healthy, you have no immunologic memory of COVID-19, you probably have some cross-reactive memory because you've had other coronaviruses, and you encounter this virus, and generally through the upper respiratory tract, and we are met by our first level of defense, and that's the innate immune response. And I'm going to talk a lot about interferon for the next few minutes as just that little piece of the story, which I think has taught us a lot. And, some people are very lucky. I always talk about the history that I hear from patients, and one strikes me very strongly, of people driving in a car with one person that had COVID-19. They're on a long road trip, and there's 4 people. And at the end of them, 2 have caught it and 1 hasn't. And it wasn't because they had, a vaccine or they had been infected before. They had breathed in that virus for 6 hours, and they didn't get it. So there are elements that can stop this in its tracks before it actually engages adaptive immunity. But for most of us, we get this, and the virus then proliferates. We become enriched for this, particularly in our respiratory tract. And within 5 to 7 days, we've gone to stage 2, and that's triggering adaptive immunity. And we make this robust antibody response. And within 10 days, you have all isoforms of antibodies present, MGA. And you are now mounting a CD4 response. 100% of people will have detectable CD4 memory responses at the end of the day. And most people are making a robust CD8 response as well. And, if you go from 2020 to now 2023, we used to say on the top here, but when we first published this in 2020, that it was severe in 15 to 20 and fatal in 1 to 2%. Today, with Omicron and with our immunity, either through vaccine or previous infection, the number of people that go into the hospital, even among high-risk patients, is less than 1%. And so it has changed for the moment. And the risk factors for this are noted. The greatest risk factor is age. After the age of 5, the rate of mortality, at least in pre-Omicron, doubled every 5 years through the age of 80. 4 logs, something that can't be escaped. Now we're focused on immunocompromised, and that's for tomorrow. So what triggers this immune response? Foreign antigen was the theory of Sir Frank McFarlane Burnett. The forbidden clones won the Nobel Prize in the 60s. Infections have always been considered to be a leading stimulus for the immune response, but now in the era of we recognize ourselves as functioning holobionts that are infested and covered with infections. That is not an adequate explanation. And the danger hypothesis has been with us for over 25 years and seems to answer many of these features. But we're now kind of thinking of immunology as having a much broader view. And those dangers can be just dysmetabolic, oxidative stress. Our immune system is triggered in people with type 2 diabetes who have no infections, this and that and the other thing. So it is far more refined than we had given it credit to. The notion that primary drivers of the immune response are pathogen-associated or damage-associated still is highly viable. PAMPs and DAMPs, and there are receptors on all of our cells that can recognize these signals to turn on the immune system to ultimately defend us and protect us. But this is now the new frontier in immunology, and I really believe that the last 3 years this has shown us to be the case, and I've talked about this extensively just over the past 3 days here, that dangers do not have to be PAMPs and DAMPs. And these dangers may be in much different forms that even 10 or 20 years ago we had no idea of. And our immune system can be triggered by what we now call neuropsychoimmunologic triggers that may be from exogenous threats, changes in the atmosphere, global warming, social determinants of health, living in pollution. It may be intrinsic, and this is striking. On the way out here on the plane, on Thursday, there's a news piece in Nature this week that kind of overstated, and I tweeted it. It said, your brain can help repair the tissue damage. And it's a beautiful story. It's about a 10-minute read. I recommend it. Our brains are intimately tied to our immune system. It's a two-way street. There's communication between the immune system and the brain and the immune system. We're now in the world of immunoautonomics. We're involved in, I know that clinical trials of vagal nerve stimulation are going on out here in Arizona, involved in others ourselves. This is a new horizon and a new frontier for this. So we have to think about this, is that existential threats can modify our immune system. And I think that tomorrow when I talk about long COVID, this is really the area that we're so interested in. And that doesn't mean that it's all in your head. It does mean everything is all in your head, but in an intense biologic way. I like this figure. I tell all of my patients the brain and the immune system are one organ. I say that the immune system is the seventh sense of the brain, and that if you want immunologic health, it has to start at the top. All right, with that story as an introduction, because I think it's a good story, this is how we still organize our immune system. We think of it in a compartmentalized way of anatomic and physiologic barriers that are highly alive. COVID-19 has shown this, that releasing defensins in our respiratory tract are hugely important to protecting us from incident infection. We have mucociliary barriers, physical barriers, acid barriers that protect us. But underneath that, we have this elegant immune system that we artificially divide into innate and adaptive immunity, which are highly integrated. And actually, there's a paleontologic history of this, which I'll refer to a couple times during this. This is the phylogenetic tree. That little red dot on the right is vertebrates. The rest of this is life on our planet as we know it. Below that dotted line are prokaryotes, and everything else is defended by innate immunity. So I was giving a talk to a bunch of fellows at a meeting, and somebody said, would you rather have an innate or adaptive immune response if you had a choice? I said, oh, innate, for sure. I said, the cockroaches are still around, and they're not going anywhere. And they don't have a T or B cell, and they're working just fine. But it just shows you how refined we are, and why vertebrates have required this. Is a matter of hand waving and white wine drinking. It has to do with longevity, complexity of biology, but that's the way that we're organized. The innate immune system, we used to say it was all defenses without memory. We now have the notion that actually innate immune system is associated with trained memory, and that just triggering of innate immunity can protect downstream from multiple pathogens. It is an early response, and the whole idea is, protect the host without damaging it. So if you, stick your finger on the rusty nail while you're working in the backyard, you want to heal this thing in a couple days. You don't want to trigger T cells and B cells. You want to get better with this. And so there's a lot of tools in the toolbox to do this. Unlike our adaptive immune system, the receptors and modulators of danger are germline encoded and do not have the vast diversity of adaptive immunity, which is generated through mutations. So it has a very broad brush for recognizing danger. There are a number of cellular elements. I always have to just kind of do this rundown. This is all the cells that are, kind of involved. This is the way we used to think of immunology. Here's some lymphocytes, and here's some cells that of innate immunity, and they're separate, but in actuality, there's intense cross-talk about this. And let's just pick a few of them just to make sure. Dendritic cells are those cells that are sentinel. I like the analogy that they are wine tasters. They are located at mucosal and barrier surfaces. We have some that circulate in our blood, a few percentage of our cells. They are divided into myeloid and plasmacytic. We'll talk a little bit about those in plasmacytic in a moment. And they determine whether threats are dangerous or not. They don't kill anything to any certain degree. And once they have detected danger, they are able to migrate, and they migrate to secondary lymphoid organs to recruit cells of adaptive immunity. The Nobel Prize for a lifetime of work was given to Ralph Steinman for their elucidating their behavior. Monocytes and macrophages are part of a single lineage, and now in the world of single-cell biology, we know that they are clouds of cells with different functions that can be polarized. Macrophages are immobile cells that have differentiated in varying tissues, and the macrophages in your brain, these microglial cells versus the cells that are located in your liver, of like Kupffer cells, differ a lot, but they are potential, they're sources of inflammation and cellular killing, but they serve different roles in each organ, and they can be differentiated into many different types of subtypes. Neutrophils, when I was doing my postdoctoral work as a young guy, no one would study neutrophils. It was like there were very few laboratories that had any interest in them, and now this is totally a paradigm shift. The notion that neutrophils die through different mechanisms. They explode, and rheumatologists know that leukocytoclastic death, and that's associated with inflammation and tissue damage, and we see it in vasculitis. There are billions of cells that die every day in an orderly fashion of apoptosis, and that is how the garbage should be taken out. But there are many other forms of neutrophil death, pyroptosis, and netosis. And netosis is when the DNA of these neutrophils is decondensed and is extruded in the cell, and the proteins undergo secondary modifications and drag out all the contents of that neutrophil. Enzymes, PR3, MPO, involved in vasculitis. Proteins involved in coagulation. Nets have been incriminated in the pathogenesis of COVID-19, particularly in the hyperinflammatory and immunothrombotic stage. Beautiful work by Jason Knight and others in the field of rheumatology that have made major contributions to this. Nets are toxic to endothelial surfaces and are highly incriminated in the pathogenesis of lupus, where these low-density neutrophils are rich sources of this and serve to both stimulate interferon and propagate damage. And then finally, this whole new fear, if you haven't gone to medical school in the past 5 years, this was not part of your immunologic training, in this emerging field of innate lymphoid cell biology and gamma delta T cells, as well as mate cells and ILC cells, playing a major role in the immunopathogenesis of psoriatic arthritis and spondyloarthritis. And these cells have also been incriminated in the pathogenesis of COVID, as they are rapidly destroyed as we move forward. PAMPs and DAMPs, pathogen-associated danger signals and danger-associated signals that are generated from our own cells have the capacity to serve as the stimuli for all of these cells of innate immune system, and some viscerosomatic cells like endothelial cells. This is a very hot area right now in immunobiology, the endothelial immune axis. Endothelial cells make cytokines. They are a source of danger for chronic low-grade infection. And they are involved in both inflammation, tissue repair, and ultimately can lead to adaptive immunity. These danger signals come in many different flavors. If you're a lupus person, you're focused on nucleic acids, RNA and DNA, and particularly nucleoproteins, is very important stimuli of the interferon response. Infections can provide danger signals as well. And then endogenously, we're rheumatologists, man. Nothing like seeing the uric acid under that microscope. That is a very potent stimulator as well. So the signals are there, but how are they detected? Well, we have developed on an evolutionary basis all of these receptors that have the capacity to be triggered by danger and then to function in terms of defense and repair. It's such an interesting story that it's really only been discovered in the past 30 years that this is how this works. I mean, this story of immunology is evolving with all these different components. And, we knew about T cells and B cells, but, how this actually functioned to turn on our immune system. Historically, I give the greatest credit to Charles Janeway, who was an original thinker and bucked the prevailing theory of turning on the immune system, and unfortunately died as he was instrumental in discovering toll-like receptors and their function preclinically. Should have been awarded the Nobel Prize, but because it's not awarded posthumously, was not. And others, including Bruce Beutler and Jules Hoffman, went on to receive that. So toll-like receptors now are, part of the common parlance when we talk about the immune response. And these are receptors that are expressed by a wide variety of immune-based cells, both on the cell surface and on endosomal, on intracellular compartments that are designed to detect danger from a wide variety of insults: viruses, bacteria, damage-associated proteins. And once these are triggered, the question is, what are they doing? Well, they have the capacity to turn on the inflammatory response. That is the signal. It's dangerous, turns on inflammation. It's not dangerous, no inflammation. That's the way it's supposed to go. This is a more detailed version of it. Once toll-like receptors are triggered, they have the capacity to do one of two things, or both. They can generate the inflammatory response via the NF-kappa B pathway, which is now being targeted by multiple therapeutics, or alternatively, some of them, particularly those that are triggered by nucleic acids, have the capacity to turn on the interferon response. And interferon has been just a hotbed of biology and biologic interest during COVID-19, because we know that interferon is important in preventing us from a wide variety of infections, particularly of viral nature. So now let's talk about the story of interferon. And again, let's go back to the paleontology of the immune system. Here we are at the top, the Vitruvian man, and here we are 700 million years earlier in the era of multicellular organisms, only with an innate immune response. And it wasn't until about 500 million years ago that we started to see the evolution of more complex organisms, ultimately dating this back to this fish, which you can see its jaw jutting out there. It's called a gnathostome, a jawed fish. And for whatever reason, this has been kind of the touchstone for identifying that point in evolution where we start to develop adaptive immunity. Lymphocytes, jawed fish. And on the left is the same point in time where evolutionary immunology has identified the rudiments of encoding interferon. So as we got more complex, we developed these lymphocytes. We also developed an interferon response. Now, the world is crazy, you know, it's going on for billions of years. Life has been around for probably 3.5 or 4 billion years of our planet, almost from the very beginning. But as these prokaryotes and viruses started to evolve, they would attack anything that was also in its evolutionary phase. So in a Darwinian term, there's a battle raging. Predator-prey. And, you know, we reproduce, we variate, and then we select. And, you know, Darwinian biology is you know, it's amazing it's still under question. You know, people say, well, isn't it only a theory? It's like, it's not a hypothesis. It's how life works, and so does immunology. And so I like to think of this as like a Peloponnesian War. So the viruses, which constitute 10% of our genome, at least endogenous retroviruses, have been battling us for, you know, hundreds and hundreds of millions of years. And so the viruses have been developing defense mechanisms to parse our complex immune response, and we have been trying to stay on top of this. So enter SARS-CoV-2, a totally new virus, you know, 3 years ago. And it's coming on to the battlefield here, and these are our weapons. And, you know, what is the story here? You know, how has it parsed and penetrated our defense to the fact that there are now billions of people that have been infected with this and millions of people that have died from this. Here's the virus. Everybody knows it. It's coronavirus. Those crown structures. And here is the virus. And I actually took this from that same New England Journal article from a couple of weeks ago. It's very nice. And it shows the virus penetrating a cell. And everybody talks about, you know, ACE2. That's the receptor that can lead to the virus entering into the cell. But viral breakdown products, including its RNA, its protein, and nucleoproteins, can also trigger these danger receptors on the cell surface. And here you can see it engaging multiple toll-like receptors, particularly those that recognize RNA. You see that once it enters the cell, it can disrupt the intracellular homeostasis, and it can turn on the inflammasome. Inflammasome, well known to rheumatologists, being able to generate interleukin-1. We can see that these proteins can also react to other toll-like receptors like TLR3 that don't recognize nucleic acids per se. All of this can start to generate the interferon response and inflammation. And that choreography of the inflammation is so important in how we exist. So we come back to this. So here we are. Now we're in Omicron, doing good. You know, 95% of the population has immunologic memory to SARS-CoV-2. In the United States, 80% of people are vaccinated. Well over half of people are infected. There are very few people that don't have any type of memory for this. But from the beginning, a small percentage of people go into phase 3, and they still do every day. And phase 3 is an unbridled hyperinflammatory phase that is still being figured out. You know, at the present time, there's over 350,000 articles in peer review that have been published on COVID-19 in the past 3 years. And there's another 50,000 in pre-publication format. There's approaching 10 billion Google citations. That's the gray literature. Much science comes out of there. So nobody really knows what's going on. It's like looking at that constellation. We see who's pointing to the bright stars. But the stage 3 is still a very important and enigmatic and instructional phase of this disease. And tomorrow, Pooja Mehta from Imperial College, did I get that right? One of the colleges is going to talk about, she's kept track of this so brilliantly, and I look forward to hearing this, an update on this. So here we are, antiviral immunity to protect us from going to this stage 3 at 30,000 feet. I've shown you that all the defenses that can turn on the interferon response. I told you that interferon, once triggered, can provide a broad antiviral defense and also can instigate and choreograph the inflammatory response. And if it is dysfunctional, we wind up with, this term that is now a colloquialism, a cytokine storm. Randy Crone, great rheumatologist, pediatric rheumatologist from UAB, had written the only book on the cytokine storm before COVID-19. And he's now got a second edition coming out. I don't know how it's going to be still up to date, but we're struggling to put our chapter in it right now. Everybody knows that cytokine storm is a clinical physiologic state that results from an unbridled and dysregulated immune response. We see it in so many settings. Everyone familiar with this with sepsis, that is, one of the early versions of this. We also know that HLH, hereditary inborn error of immunity, or children are born with immune systems that are constantly turned on, and they're cellular killing mechanisms that will die of hyperinflammatory disease unless they're transplanted before the age of 1. In rheumatology, we now talk about MAS, kind of a secondary form of HLH. We see it in lupus, we see it in JIA, we see it in adult Still's disease. Why some people get it, why some people don't, we don't know. It's our triggers. Kawasaki's, Miss A, Miss C in the world of COVID-19 are forms of hyperinflammatory disease. All of them develop multi-organ dysfunction, and while they're highly related, they are distinct. The most recent and the kind of the talk of the day now is CAR T cells, and CAR T cells are transforming the treatment of refractory lymphoproliferative diseases, and in the past year have entered into the armamentarium of rheumatology. And in May, Georg Schett's going to visit us in Cleveland and keynote our talk on CAR T cells at our biologic summit. And one of the downstream byproducts of CAR T cells, these immortal cells that last a long time, that do all this killing, is to develop cytokine storm. So this is now, something that is far, more widely understood. It's the evolving story of immunology. So if this was the model, adaptive triggering, or innate triggering, adaptive immune response, and then dysregulation, this has guided our therapies. This is like an ancient slide. But we knew that if you could pick up COVID-19 early, you'd want to treat with antivirals. And we made this slide before there were any antivirals. And now we have, a rich collection of antiviral therapy. But they have to be used early before this immune dysregulation goes awry. Wouldn't it be interesting to treat with interferon? And unfortunately, type 1 interferon trials have been really disappointing. But recent RCT of type 3 interferon, lambda interferon, which is loco-regionally active, have been positive. And, maybe that'll serve a role, maybe it won't. Cassie will talk about the ups and downs of monoclonal antibody therapy. As I say, this was an old slide. We could have put the X through that right now. But they're coming back quickly. And we have great technology to produce these in a rapid mechanism, and if the regulatory agency and the FDA could creatively create a fast track, we would have them right now to protect our immunocompromised patients. And at the other end, once you get to the hyperinflammatory stage, the greatest development and greatest advance in the treatment of bad COVID-19 is a drug that we've had for over 75 years. Dexamethasone has transformed and saved more lives than all other drugs to begin with. We now use JAK inhibitors. We now use IL-6 inhibitors. There's positive data for TNF inhibitors and abatacept. Pooja will talk all about these. So it's depending upon the balance. Early on, you want to bolster the defenses. Later on, you want to use the anti-inflammatory. That's the story of how it goes. So this interferon, let's just kind of close off this discussion with our thinking of interferon. What's time? How much time would we? I don't have a timer up here. What time is this supposed to end? 15 minutes. All right, perfect. All right. This is 1957. That is Jean Lindemann and on the right, and Alan Isaacs on the left. And these guys were very creative scientists. And they explored a clinical observation that we were talking about just the other night that is quite remarkable. And so now I think this is the 50s. They said, viruses are very common. And they worked at a place in the United Kingdom called Mill Hill, which is the laboratory that discovered influenza. They said, viruses are all over the place. We don't know that much about all of them. But it seems pretty weird that people don't have two viral infections at the same time. You don't get measles and chickenpox, and you actually don't get influenza and COVID, considering how ubiquitous these occurs, but very unusual. And, you don't get rhinovirus and coronavirus at the same time. So they said there must be something going on. So I have to fast forward this. They developed an experimental system. They're culturing viruses on egg membranes, and they exposed the egg membrane to a virus, and they tried to reinfect it, and found that they couldn't do it. The culture system had seen one virus, they couldn't infect it with another virus. They said, well, it must be something in here. And what's the mechanism? And they tried to harvest the protein. They had the hard as hell time doing it. And this is before the dawn of protein chemistry. It took them years, and they finally got this little bit of something. They said, I think we found this. And they published this data, and they said, we have this protein. It can block the infection of a culture system from another virus, and we're going to call it interferon, because it interfered with the capacity to infect this. Well, they couldn't characterize its molecular structure, this, that, or the other thing. They knew that they could degrade it with enzymes, but people didn't buy it, and they got a lot of blowback. And over a couple of years, actually, Isaacs became very depressed, died at the age of 44. Kind of a tragic story, but it got on the map. And then over time, fast forward this, and there's a great, I'll show you a book at the end that I recommend that has the backstory of this. People said, yeah, this is really a real deal. And it has something to do with viruses, and, if we give this to other preclinical models, it looks like it has an effect on certain forms of cancer. And by the 70s, this was on Time magazine. They kind of forgot about the viral thing, and this is going to be the new cure for cancer. And high-dose interferon was available, and as you know, it never met its greatest expectations. And that's where the field lingered. Nobody talked about interferon in lupus, and nobody talked about interferon in autoimmunity. That was not even a concept. This is just showing very nice article by Crow and Ron Blom, who've done so much to help us understand interferon and lupus, showing that, interferon's an early defense mechanism. When you get a viral infection, you want it early. You don't want it late, because it causes a lot of inflammation. So let's move this up to COVID-19. So much of some of the most interesting work has come from our colleagues who do inborn errors of immunity. This is an international group led by Jean-Laurent Casanova and hundreds of other investigators. They said, patients with influenza, we have influenza on the planet. Most people do okay with it, but occasionally young people die of influenza. And it's tragic when it occurs. And it had been known for some years that many of these people have genetic defects in interferon response. And so they said, well, let's look at some of those genes that have been associated with fatal influenza in young, healthy people. And these are the genes, and let's just say that they're involved in interferon pathways. Let's look at patients dying of COVID-19. And they found, because, while COVID, when it first came out, it was a disease of old people, there's always somebody in their 20s or 30s that succumbs to this. Like, what's that all about? They look very healthy. And they found that about 1%, barely 1% of people had defects in these interferon pathways that protected them. They said, well, that kind of makes sense, because interferon is there to defend us against that. But it was a rare event, and it was done. And then the next step. There are a group of diseases that we don't talk much about in rheumatology, although in 2022, Steve Holland gave a plenary talk at the ACR, because he's pioneered this whole area of immunodeficiency equivalence, where we make antibodies to our cytokines. That's not good, Well, it's not good when you make too many of them. It actually winds up being a very, very common phenomenon in biology. We make them all the time. And there are diseases that range from infections to autoimmune to other forms of disease that are driven by these natural anti-cytokine antibodies. And here's just a list from a recent review from the Holland group from a few years ago, but this list has gone along. So pulmonary alveolar proteinosis, a disease that's been known for a long time, is incriminated and driven by antibodies to GM-CSF. There are rare cases of people that make natural autoantibodies to anti-IL-6, kind of interesting, and they generally develop serious bacterial infections. There are many other clinical phenotypes from making these. And if you screen blood donors, there is a huge number of people with very tiny, low-level antibodies to multiple cytokines. So interesting. What does that have to do with COVID-19? Well, there's a disease called APS, or air deficiency, where it's a genetic defect that compromises our ability to cull our T cells from autoimmune, from generating autoimmune cells. And children are born with APS, in this air gene, which causes expression of natural proteins that allow this negative selection. These people make tons of autoantibodies, and they make tons of autoantibodies against cytokines, and among them are anti-interferon. Children with APS who get COVID do terrible, and high rate of death. I said, well, that's a very rare disease. Maybe the anti-interferon antibodies had something to do with it. And then probably the most impactful paper of the first phase of the pandemic was recognizing that as we get older, a large percentage of people make autoantibodies against interferon. And in some of them, in the pre-Omicron era, led to severe outcomes. And it has told us a lot about this condition. So now we're in the world of, trying to attack interferon from a number of different ways. We want to give interferon to stop this. We want to neutralize interferon for certain diseases. This is a timeline of the history of interferon and lupus, which, has told us that interferon is yin and yang. Too much of interferon is bad, not enough of interferon is good. Not enough interferon is bad from protecting our host defense. And the story of interferon and lupus dates back at least 40 years. Before that, it was not known that interferon could even cause inflammation. I think Ian Grosser and Bellevue in preclinical models of in mouse models showed that interferon could actually cause an autoimmune disease. Up until then, it wasn't even on the plate. Cancer, infection. We now know that the interferon signature, which you can see on the right, is present in 80% of patients with lupus. That interferon is a driver of inflammation. It's probably an upstream stimulator of this T-cell, B-cell co-stimulation and leading to loss of tolerance. And now that we neutralize this with antibodies such as anifrolumab, it's important to recognize that interferon has multiple redundancies, type 1, type 2, and type 3. When we use a drug like that, we attack type 1. And it does not eliminate interferon. It is trying to tamp it down to a normal interferon response. And that's why it is not as scary in the world of COVID-19 as we had originally feared. We also believe those patients should be adequately immunized. Finally, interferon, when it is awry, may be contributing to autoimmunity. I'll talk a little bit more about this tomorrow when we talk about long COVID. But this is a very recent paper, again, just from this month, looking at autoantibody signatures in patients recovering from COVID-19. So as you can see here, the more yellow you have, and these are looking at only of ANA specificities. This is what made this paper interesting to me. So here's a control population, no COVID-19. Yeah, some people have ANAs, we know that, 20% of young women. This is people with COVID-19 that are POSI symptomatic, and looking at them over a 12-month period. There's a few more developing ANA subspecificities. These are people that had mild to moderate COVID. These are people that were in the hospital, and these are people that were in the unit. Look at what happened. These are our tests that we use. Is highly enriched responses even 12 months later. There are data in here that show that the slope of this, and these tend to disappear. There are some other data, on the other hand, which I won't have time to show, from P.J. Utz at Stanford, that shows that autoantibodies are generated not only after SARS-CoV-2 infection, but can be seen after other infections, and even in people who have been in the unit for non-infective causes. They develop autoantibodies as well. So this is some thing for again. This is a hand waving and white wine drinking. Why are we making all these autoantibodies? I think that my thing. I'm looking over at Dr. Looney here. I think that this is somewhat suggestive that that this is a network. This might be a Niels Jerne type of notion that we make these autoantibodies. We're recognizing this. 3 years ago, we didn't recognize this. As part of our natural response to severe illnesses. Are they good? Are they driving long COVID symptoms? We don't know. Are they just, passengers in the car? We don't know that either as well. So my last slide is, here's the last story. It's interferon. It's your friend, when it's functioning as an antiviral. It's a foe when it is dysregulated. You don't want to eliminate interferon because we need it, but it has to be strike that balance. And so anyway, this year, this was kind of a more philosophic approach. I was just feeling philosophic after coming out of the first live meeting of 3 years. And these are the books that are still, I think, are pretty good. But the best one is this beautiful Cure. Dan Davis, he's a very creative guy, writes a lot of very nice things. And he tells the backstories of people like Charles Janeway and Ralph Steinman and some of the other contemporaries that we have. So I'm gonna stop here and just open this up for philosophic questions only. We have a minute or two before we go on. So thank you very much.